11, 12. Okay, everybody's here. Let's start our 11 a.m. plant cell tour. Please keep all hands and feet inside the Shrinkle bus at all times, unless you want cytoplasm all over you, which I'm pretty sure you don't. And please feel free to ask any questions at any time during the tour. Let's begin, shall we? Oh, what's that, sir? What's the Shrinkle bus? Well, this amazing contraption is capable of shrinking itself and everything inside it smaller than the size of a plant cell. In fact, on this tour, we'll be going inside a plant cell. Let's continue. Brace yourself, passengers, as I now hit the shrink button. Whew! We've landed just outside the cell. Now that we've shrunken down, let's talk about the cell membrane. If you look close enough, we can see that it forms the boundary of the cell and acts as a selective barrier that only allows certain materials to pass in and out of the cell. It's made up of a lipid bilayer that is in itself made of phospholipid heads and tails. It is also made of many large integral proteins. There are two ways of passing through the cell membrane: active transport and passive transport. Active transport is a way of transportation that requires energy input from a cell and enables a cell to move a substance against its concentration gradient, or against the gradient. Passive transportation is a way of transportation that requires no energy from the cell at all. Let's start moving through the shell, shall we? Yes, ma'am? What are we moving through? Oh, that's what I mentioned earlier. Cytoplasm. It is the entire region between the nucleus and the cell membrane. Speaking of the nucleus, there it is on the left. As you can see, the nucleus is very big. Let's stop by for a closer look. The nucleus. It contains most of the genes that control the cell. It also contains the nucleolus and chromatin. From our view, you cannot see the nucleolus or chromatin, but trust me, they're in there. The nucleolus is where the components of the ribosomes are synthesized and assembled. What are ribosomes? I'll get to that in a little bit. Chromatin consists of DNA and protein and then condense to make chromosomes. Outside the nucleus is what's called the nuclear envelope. The nuclear envelope is the double membrane that forms the boundary between the nuclear contents and the cytoplasm. If you look close, you can see that it is perforated with pores. Look to the right, everyone. There's a free ribosome. There are in fact two types of ribosomes. Free ribosomes, which we see on the right, which are the site of protein synthesis and produces proteins for use within the cell. There are also bound ribosomes that are attached to the endoplasmic reticulum, or ER for short. Bound ribosomes are also the site of protein synthesis, but it produces proteins to be used in the outside of the cell or in the cell membrane. Bound ribosomes are called bound because they're attached to the rough ER. There are two types of ER in the cell: rough and smooth. The rough ER is what kind of surrounds the nucleus. Its job is to channel proteins to transport vesicles. I'll talk about vesicles later. Let's keep on moving to the other ER, the smooth one. The smooth ER on our right is called smooth because, unlike the rough ER, it doesn't have ribosomes attached, so it appears to be smooth. Now let's make our way to an organelle that separates plant cells from any other cells like it. On our left is a chloroplast organelle, my favorite organelle. This organelle is what gives the plant its green color. It also is the site of photosynthesis, and it produces food using light, carbon dioxide, and water. We pass another organelle on our right, the mitochondria. The mitochondria. It is known as the powerhouse of the cell. It is the site of cellular respiration and produces ATP from sugars, fats, and other fuels. We can also see on our right the Golgi apparatus. The Golgis consist of flat membranous sacs. They also receive transport vesicles from the ER. Speaking of the devil, if you look closely, you can see a transport vesicle entering the apparatus. There are two types of vesicles: transport and secretory. The transport vesicles, as you can see, only transport materials to the Golgi apparatus. Secretory vesicles carry Golgi modified products to the cell membrane. And now, the largest organelle, the vacuole. The vacuole is a very large, membrane-bound sac that stores materials. Who here has forgotten to water their plants from time to time? And they get droopy, right? Well, the reasoning for that is vacuoles in plant cells are used just for water, or mostly for water, and they help hold the plant up straight. But when you forget to water your plants, the vacuole must use its own water to help the plant grow. But because the vacuole isn't in its completely upright position, it slowly droops down. So remember to water your plants so your vacuole doesn't have to do all the work for you. The last organelle on our tour is the cytoskeleton. It is made up of microtubules and microfilaments. Microtubules are straight, hollow tubes made of tubulin. There are also microfilaments. Microfilaments are the thinnest filaments of the cytoskeleton, and they are found in the cytoplasm.
Well, that was the last organelle. Hang on while we dilate to our original size. Before we exit the shrinkobus, I want to talk about one last thing. Cell theory. If you take one thing away from this tour, please let it be the definition of cell theory. Cell theory is the idea that all living matter consists of cells. Cells are the structural function units of life, and that all cells come from pre-existing cells. Cells are the basis of all life, and it seems that we take them for granted. So thank you very much for taking the time to learn about the organelles of a cell plant. Bye! Randy Orton wishes you a safe journey home.